goal of this week's lab is to examine cells. We're going to use the microscope to examine cells, and we'll be looking at various microscopic techniques to examine a variety of different cells. This is the microscope we'll be using. This is a brightfield microscope. Brightfield microscopes begin at the bottom with a light source. The light source shines light up through the substage. There's a condenser that basically focuses light. The light passes through an object, then goes directly through. It's transmitted up into one of these structures, which are called objectives. It then goes through this tube, bounces off a mirror, hits the ocular, and it's magnified again by this lens, which is a 10x ocular. Here we have various objectives. We have a 4x objective now in place. We always want to start with our lowest magnification in place because then it's easy to move things on and off the stage. We have a 10x objective, a 40x objective here, a 40x objective here for something called phase contrast, which we're not going to look at, and a 100x objective up here. Okay, so our 4x objective is in place. If we have a 10x ocular, a 4x objective, 4 times 10 is 40x. That will magnify a slide by 40x times. Our 100x then is our maximum. Our 100x is designed to be used with oil. We will use the 100x at the end when we look at bacteria. That 100x then times the 10x ocular is 1000x. Now we also have a camera attached. This camera then goes right into the eyepiece. So we basically take this eyepiece out, put this camera in, and we have it hooked up to a computer. We can get a live image of the computer. So everything that I see in the microscope, you will see on this computer image, and you will get a nice live view of exactly what's going on with the microscope. For example, here's a slide that I made up. This will be one of the slides that we'll be looking at later in lab. We'll just throw it in here really fast so that you can see what's going on. And we'll focus it, kind of move it down a little bit, put some stuff in the center that looks cool. Then what I can do is easily switch to a higher magnification, focus it. Okay. Center the object. There's actually several things on here which are pretty neat. I just have to choose one. Center it, focus. Alright, then we could center it again and look at various objects. So here. The first technique we want to do is a wet mount. To do the wet mount, we're going to use it to look at plant cells. So our plant cells are going to be off of the leaves of this plant, which is Elodea densa. And first step then is to take a Kim wipe, make sure that my microscope slide is totally clean. So I'm going to clean off that microscope slide really well. Then I'm going to reach in here. We want a teeny tiny leaf off the top. So you get a little leaf right off the very, very top of that. And I just want one leaf. We'll put it down and stick it down on there. And now I'm going to take my eyedropper, get a drop of water, put that right on top there. Now I'm going to grab a cover slip and gently put the cover slip, whoop, we just want one cover slip, not two. Gently put the cover slip down on top. And here is our wet mount all ready to go. Next we want to look at human cheek cells. So in order to look at human cheek cells, I have a variety of items. I have a microscope slide. I have toothpicks that I'm going to use to get the cheek cells off of my cheek. I have a stain called methylated blue. I then have a staining tray that we're going to use to stain the slide, and I have a Bunsen burner to do what's called heat fixing the slide. So the first step is to basically take a drop of water. We'll put a little teeny tiny drop of water on the slide. I'm then going to grab a toothpick. I'm going to rub it on the inside of my head. So all on the inside of my cheeks, I get all kinds of cheek cells on that toothpick. Now I'm just going to then take the toothpick, circle around a nice big circle there, so that it quickly evaporates on that slide. Okay. Now as it's evaporating there, I'm going to just set it up here on the staining tray. Now that my slide is dry, it's time to do what's called heat fixation. Heat fixation is basically taking the Bunsen burner, allowing heat to go on the bottom of the slide so that the cells stick to the slide. Later on, I'm going to add a little bit of methylene blue, then we're going to wash it off with a wash bottle, and we want to make sure that the cells don't slide off. So to start with, I'm going to light the Bunsen burner. Okay, we'll get a nice flame going. You see that nice cone on that Bunsen burner there. I'm now going to take my slide. I'm going to just go over slowly twice over this slide, counting 1 Mississippi each time. I'm going to angle the slide away so when the flame hits it, it goes that way. You know, a lot of people use clips so that they are a lot farther away from their fingers. So we're just going to go 1 Mississippi, 1 Mississippi, and put that there. I'm going to take my methylene blue and cover the slide with methylene blue. And carry this smear with methylene blue. Alright. Let's smear that. We'll get covered with methylene blue. We're then going to wait for a minute, and after a minute we'll wash it off. The methylene blue has been on for 1 minute, so I'm going to wash off the stain. So I'll just take my wash bottle, my slide, angle it down, and just simply wash all of that methylene blue off. By heat fixing over the heater, then my cells stayed on when I washed it off. Now all I have to do to finish this off is just basically blot it dry with Kim wipes, and then I can look at it underneath the microscope. To go under oil on the microscope, what I'm going to do is I'm going to swivel halfway between my 40x and my 100x, put a drop of oil directly on the microscope slide, then swivel in my 100x. This is my oil. I'll open my bottle, swivel this halfway between those two, and then put a drop right on the microscope slide itself. Now I'm going to swivel in the 100x. Once I'm under oil, I can never go back to the 40x because this is not made for oil, whereas the 100x is. Oil is designed to make more light. As you increase magnification, you need more light to increase the resolution to actually be able to see a cell. So we need that oil on there to have enough light to visualize those cells underneath the scope. So the technique we'll be working on right now is just simply how to put away your microscope. So here we have a microscope. It's in focus. We've got a slide on here, oil immersion objectives in place, and it has oil on it. And so now I want to put it away properly. And so key thing is if there's oil on this slide right here, I can't swivel back to the 40x because this 40x is going to go right in the oil. So I have to swivel my scope this way. You notice I did not change the focus. I just swiveled the 4x in place. That gives me room for the stage to where I can go in and I can remove the slide. Now what I want to do is I want to clean the objectives. So to clean the objectives, I do not want to use Kim wipes. Kim wipes are in here to clean off slides. Okay, these are never to be used on the objectives because these can scratch the objectives. So slides only, not objectives. In the room then you will have little containers called lens paper. We've cut the lens paper into little squares for your convenience, and so we can take the little square and we can see what's happening. Alright, so now I'm going to actually swivel this one more over to the 10x because the 10x still has a lot of space. And I'm going to go over here to the 40x. I'm going to wipe that. I'm actually going to swivel it so I can reach it. I'm going to wipe that 40x. And if we look at the lens tissue on the 40x, it has a teeny bit of oil on there. So whoever used this scope last left a little bit of oil on that 40x objective. That should never be the case. I should never have any translucence from the 40x objective. So I'm going to wipe it again with a new piece, and you can see that's better. I'm going to take a third piece that has no translucence on it, and we will wipe it a third time, and then that objective is totally clean. Now, if we go over to the 100x objective, we have a lot of oil on the 100x objective. So I'm going to swivel it back over here. Here's my 100x objective. And if I wipe my 100x objective, okay, we then can see all that translucence there of the oil on my 100x objective. So I'm going to want to go back and wipe it again. And really it might take 2 or 3 times with a real clean piece of lens paper where I wipe in various clean spots. I might shift it and go to a nice clean spot. And then I wipe it. I grab a brand new piece, wipe it, and if I have no translucence at all, it's ready to go. Then what I want to do is wheel in the 4x. Do not change the focus, because that means the next student comes in. If that student just goes and puts their slide on the stage right there, this will now be in focus. I don't have to crank this up, crank this down. It's in focus. The next student is in focus. If everybody does that, we don't have a problem focusing, because one of the biggest time sinks in here is focus, and the big problem there is because students crank this up and crank this down. Put in the 4x, remove the slide, and you are set. And then the last thing that I want to mention is that for a lot of students, one of the things that happens is that they'll take a piece of lens tissue, they will wipe the 100x objective and then get oil all over that lens tissue. And they'll basically take an oily piece of lens tissue, go over here, wipe the 10x, and then in effect wipe oil onto the 10x and oil onto the 40x. Always make sure you have a very clean piece of tissue so that you never wipe oil on an otherwise clean objective.